Good morning, ladies and gentlemen of animal physiology. My name is Bo Wilkinson, and today I'm inviting all you lovely people to join us on an adventure. You probably have lots of questions like, where are we going? What should I pack? Do you know how to drive this thing? But fear not, all will be answered very soon. I'll be your guide along with Yi and Jia Cheng, and today we'll be leading you through the wonders of the stomach.
We all have a pretty good idea of the role our stomach plays in our digestive system. We know it stores the food we eat. We know that it breaks down the food molecules through muscle conjunction, but our focus today will be on how the stomach digests the food chemically, especially through the secretion of hydrochloric acid, HCl.
To understand how HCl works in the stomach, we must first understand where it comes from. Parietal cells are cells located in the gastric gland in the lining of the fundus in the stomach, and they are responsible for the secretion of massive amounts of HCl and water into the stomach. The way HCl is produced by parietal cell is through a series of chemical reactions and ion exchanges.
First, carbon dioxide, which is produced as a byproduct of normal metabolism, enters into the parietal cell via simple diffusion, and the enzyme carbonic anhydrase catalyzes a reaction between the CO2 molecule and water to form carbonic acid, or H2CO3. The carbonic acid then dissociates, forming a bicarbonate ion and a hydrogen ion. The bicarbonate ion travels to the plasma membrane of the parietal cell, where it is exchanged with a chlorine molecule through the actions of an anion transport protein on the membrane of the parietal cell. The hydrogen ion is actively transported across the membrane of the parietal cell into the lumen of the stomach with the help of a hydrogen-potassium ATPase, which is a proton pump that works by exchanging a hydrogen ion out of the cell for a potassium ion taken into the cell. This exchange requires ATP, but when the chloride ion leaves the parietal cell through facilitated diffusion, ATP is not needed. Once in the lumen of the stomach, the hydrogen and chloride ions join to form hydrochloric acid.
Already have we known how HCl is secreted into our stomach? Then we can start with how do those stuff in our stomach work together to get rid of the tough food we eat. We can digest a lot of protein in our stomach, which is done under the function of multiple elements. Thanks to the secreted acid, the protein we take in is denatured in our stomach juice, which is adjusted to a really low pH condition. Then the further digestion of protein is carried out by pepsin, a kind of proteins in our stomach, which requires an acid environment to be activated and play its role. The acid in our stomach also helps the large particle breaking down into small particles.
So we can see that gastric juice is really something, right? So how do our stomachs survive these extreme situations? The answers can be a little bit complicated. Firstly, the inside cells of stomach are well protected by a layer of real lipidity, non-permeable mucus, which shelters the cells from direct contact with gastric juice. Bicarbonate ion is secreted and trapped in the mucus, and it can help neutralize the acid in the vicinity. These ions help protect our mucus and stomach from being damaged by acid. The mucus can really help our stomach, but the stomach itself also has something special. The inside cells are held together tightly with some compounds to build an impenetrable wall to stop juice from leaking and doing harm to our bodies.
Here we would like to talk about the regulator and hormones involved in acid secretion. There are many molecules involved in this progress, and today we will introduce the most important four. And there are three positive regulatory molecules, acetylcholine, histamine, and gastrin, and one negative molecule, somatostatin. Also, we will discuss the key protein, proton potassium ATPase, the proton pump, and the whole picture of the regulation progress. All three positive regulators increase the acid secretion by promoting integration of additional proton percussion ATPase pump. When the cell is rest, these pumps are located in the vascula inside the cells. When the cell is stimulated, this vascula fills with the plasma membrane, thereby increase the surface area of the plasma membrane and also the pump numbers. The figure shows the whole picture, how the different molecules interact to regulate acid secretions. As we talked before, acetylcholine and histamine directly stimulate parallel cells to increase the acid secretions. Gastrin stimulates acid secretion by stimulating histamine release from the ECL cells, and it also has the direct effect on the parallel cells, which to increase the cell numbers. In order to keep stomach pH at a normal range, so there also are negative feedback on this progress. So when the pH of the stomach gets too low, somatostatin secretion is increased, and it inhibits acid secretion by direct effect on the parallel cells, and it also inhibits the release of the positive regulator, histamine and gastrin.